So let's look at blood type as another example of multiple alleles. This is probably the most common multiple allele example that you're going to come across. Now, A, B, and O blood types refer to the type of antigen present on the red blood cell. So an antigen is something that stimulates an immune response. So we'll talk about why these are called antigens in our blood in just a minute. But on the outside of our red blood cell, we have something called an H antigen. So this H antigen is a string of sugars that are attached to that red blood cell. Now, there is a specific enzyme called a glycosyltransferase that attaches sugars, a specific sugar, to the H antigen. So all blood types have this H antigen, but individuals who are O blood type make an inactive form of this glycosyltransferase. And so there are no sugars attached to their red blood cell H antigen. So that O blood type lacks a specific antigen. Sorry about that. The O blood type lacks a specific attachment on the H antigen. So there is no sugar attached to that O blood type H antigen. And if you look up here on the notes there, it's just indicated by an H. It's the H only attached to that red blood cell. If you are an A blood type, the enzyme, the glycosyltransferase that is produced in your body actually adds a specific sugar, N-acetylgalactosamine. You don't have to remember that. You can just call it the A antigen. If you are A blood type, on your H antigen, you have a antigen. That functional glycosyltransferase is going to add a specific sugar there. We'll just call that the A sugar, which is why we indicate it by HA. Now you might guess that B does something similar. So in the B blood type, that glycosyltransferase adds a galactose. We'll call that the B antigen. That's indicated over here by the HB. So if you are O blood type, you have no antigen attached to your H antigen arm. If you are A blood type, you have the A antigen. And if you are B blood type, you have the B antigen. So this is an example of multiple alleles. Because you have that O allele, you have that A allele that produces the A antigen, and you have that B allele that produces the B antigen. So if you are blood type A, your blood has the A antigen on the outside. Blood type B is going to have the B antigen. Blood type O is going to lack antigens. Now there's a fourth group I haven't mentioned yet, and that is the AB group. If you are A and B, if you are blood type AB, you are going to have the A and the B antigen. Now here is where we start to see some different types of dominance going on. If you are blood type AB, you are demonstrating a co-dominant phenotype because you are producing both the A and the B. So recall from our last video, the roan cattle, where you're producing both colors of fur. So A and B are co-dominant to one another. Now, if you are blood type O, you actually are recessive. You're fully recessive to both A and to B. If you are either A or B, you can be homozygous, or you can also be, heterozygous for the A allele. If you are B blood type, you can be homozygous for the B allele, or you can be heterozygous for the B allele. So we see ranking dominance where A and B are dominant over O, but A and B are co-dominant to one another. Now, the reason that these are called antigens, remember I said I would come back to why they're called antigens, is that your immune system actually produces antibodies for the opposite antigen. So if you are blood type A, you have the A antigen, which means you produce the anti-B antibody. So if you receive type B blood, your body's going to have an immune response because it has been exposed to that B antigen. Likewise, if you are blood type B and you produce the B blood with the B antigen, your body is going to produce an A antibody. So if you receive blood type A, your body is going to produce an immune response against the A blood type. Now, what if you're A and B? You might have realized this could cause problems. If you are AB blood type, you produce no antibodies. That means your blood can produce both of those antigens and not attack itself. If you are O, you have no antigens, but you produce both A and B antibodies. So keep that in mind. We're going to talk about this when we start to get to our genetics problems. All right, so this question always comes up before we get to our problems, and it has to do with blood donation. So O blood type is very often sought out by blood banks, specifically the O negative blood type. Now, the reason is that O produces no antibodies. Produces no antigens, which means it won't stimulate an immune response in anybody that it's given to. So O, especially O negative, and I'll talk about the negative in a minute, O negative is going to be the universal donor because everybody can take it, because nobody's blood is going to have an nobody's body is going to have an immune response because there are no antigens. AB, specifically AB positive, is going to be your universal acceptor. So AB produces both antibodies, produces both antigens, but it produces no antibodies. So it can't reject anybody else's blood type. So AB, specifically AB positive, is going to be your universal acceptor. And then we've got different combinations in between depending on what antibody-antigen combination you have. That the positive and the negative refers to something called the rhesus factor. Now, this is another antigen on the blood. The positive, the presence of the factor is going to be dominant over negative. So a negative individual can donate to a positive without having any sort of reaction. Positive individual cannot donate to a negative because it will cause a blood reaction as well. We can talk about this a little bit more in lab if you have some questions. You do not need to memorize this chart. I just want you to understand the antigen antibody problem. All right, so we're genetics, so let's get to some genetics problems. So if you have the A allele, you are going to produce B antibodies. When this happens, you get blood clotting because your body is producing an immune response. If you are the B blood type, you're going to produce those A antibodies. And so you get blood clotting if you come in contact with the A blood type. O allele doesn't produce any antigens, and so it doesn't react with any other blood type. Now let's look at the genotype. So if we are blood type A, we can either be homozygous A, we already went over this a little bit, or we can be heterozygous with the O allele. So the A allele is going to be dominant over O. Likewise, B, you can be homozygous B, or you can be heterozygous for the O allele. O is fully recessive. So if you're the O allele, you are going to be homozygous recessive. This should actually be little I, at least for the purposes of this class. That's how I'm going to note it. And then if you are AB, you are demonstrating a codominant genotype. So let's look at some examples here. All right, so if we have a mother who is blood type A and a father blood type AB, what are the potential genotypes of the offspring? So let's say we have a mother with blood type A and a father with blood type AB. What are the possible genotypes and therefore blood types of the offspring from this union? So let's say that the mother can be either heterozygous for blood type A or homozygous for blood type A. So she has those two options. Now let's say that dad is AB, so he is going to be codominant heterozygous for blood type AB. So there are two different potential crosses here. So if mom, oops, I wrote that wrong up here. If mom is heterozygous for A and dad is heterozygous for AB, then the possible blood types of their children are A, AB, A again, or B. So blood types, if mom is heterozygous, the children can be A, AB, or B. Now, if mom is homozygous for A and dad is heterozygous for AB, their children can be A, AB, A again, or AB. So from this one, they can be either AB or A. So depending on what mom's genotype actually is, we have all possibilities except O. So O is not a possibility here. Let's say the mother is blood type A and the father is blood type B. Again, if mom is A, she can either be homozygous for A or heterozygous. Dad is exactly the same but with the B allele. He can be homozygous B or heterozygous. So we've got lots of different combos here. Let's use our buddy foil to help make some crosses. So we can be a cross between the homozygous individuals, in which case every offspring is going to be AB blood type. We can cross the homozygous with the heterozygous B. in which case we will get AB or A. Those are our options. So we can get AB or A here. Let's do the opposite, the heterozygous for A and homozygous for B. We're going to get the opposite there. We're going to get AB or B. And then let's do the two heterozygous. So IA, IB, I. Now in this case, we get all four options. We can get AB, we can get A, we can get B, or now we can get O types. So depending on what the parents actually are, what their actual genotype is, you can get any potential offspring from here. Now, you could do a pedigree, you could do blood typing experiment to actually figure out, well, blood typing wouldn't work here. You could do a pedigree to figure out what mom and dad are, to determine if they are homozygous or heterozygous. If you don't have that information, you would have to give me all of the potential, blood types for the children. All right, let's work backwards. All right, if a child is blood type B, and both his parents are type A, can they be his biological parents? So we don't even need to work out any Punnett squares here. If the child is B, the possibility for his blood type is that he is homozygous B or heterozygous B. For either parent, if they are blood A type, they are either homozygous A or heterozygous A with the O allele. So there are no B's to pass on. So these parents cannot be his biological parents. So these parents are not biological because he could not get the B allele from any type A parents. So let's look at one more. This is sort of the opposite. If the child is type B and mom is O, what could the father be? What blood types could be the father? So the child could be homozygous or heterozygous. The mom is homozygous. So we actually know that the child can only be the heterozygote. The child has to be heterozygous because if mom is type O, she has to be able to give that O allele. So the father has to pass on that B allele. But what ways can he do that? He can do that if he is blood type B, he can pass on that B allele, or he can pass on that B allele if he is AB. So the father could be type B or type AB. He could not also be O, and he could not be A. So let's look at one more example of a multiple allele situation with ranked dominance. This is something called the agouti gene. So the reason that we talk about it here, even though we've already done two examples with the ranked dominance, is that this is a very common color in animals. So the agouti gene regulates a black pigment called melanin. So if you have the fully recessive gene for eumelanin, you produce this black pigment everywhere all the time. And so the animal is going to be fully black. If you have the any of the other alleles, you're going to block the synthesis of eumelanin. So if you are fully dominant for this, you actually produce something called pheomelanin, and that completely blocks eumelanin. So you see no black at all. Now this agouti gene has different expression patterns in different places. So you can actually get a different color pattern on the top half of the animal than the bottom half. So if you have this AY allele, you get the basically pheomelanin produced all over your body. And so you get an essentially yellow or orange colored animal. So here we've got a pug. These are all dog examples. Got that pug there that is showing that yellow color. If you have the next allele in the dominance order, the AW allele, you get some production of the eumelanin, and it is expressed only on the dorsal or the back, the top half of the animal. But you still produce, you still block a little bit of that eumelanin, and it is shown in bands along the hair. The next one down produces a little bit more eumelanin, and it is distributed on the back only. So then on the belly, you get the yellow color. So we've got that in the Gordon setter there. You can see it's got black on the top and then yellow on the bottom. And then if you are fully recessive, you produce eumelanin in all times and in all places. You're a fully black dog. I skipped over the elk hound. Elk hound and German shepherds are examples of animals that have the lighter color on the bottom and then sort of the banding pattern of the pheomelanin and the eumelanin on the top. This is we like to go over this because we'll often hear about agouti mice. Back in one of the early chapters, maybe chapter 2, there was a little box that talked about agouti mice. So the agouti gene is going to pop up in a few places. So I like to talk about it so you know what the heck that is. But again, it is an example of ranked dominance, depending on how much pheomelanin you produce to block that eumelanin in different parts of the body. All right, let's move on to another example, or I'm sorry, another dominance, inheritance pattern. There we go, that's the right word. Let's move on to another inheritance pattern. Remember, we are talking about unexpected phenotypes. Sometimes you have a single gene, one locus, that affects multiple phenotypes in an individual. So these are often disease phenotypes where you get a set of very specific symptoms, and they're often more than one. So the actual gene probably causes a very specific protein change, but it manifests itself in many different ways throughout the body. And genes are those genes may have slightly different effects depending on what cell type they are expressed in. So we have two examples here. The first is cystic fibrosis. So cystic fibrosis is a nucleotide change that results in a defective chloride ion channel. Now, that is what the actual mutation does. It inhibits cells from moving chloride ions in or out of the cell. Now, when this happens, mucus can form on the outside of the cell because those ion channels help little hair-like structures on the cell wipe away mucus that would normally form in the body. And so when you have these defective ion channels, you get a layer of mucus in your blood vessels, on the outsides of your organs and things like that. And so while you have one specific protein change due to one gene, you get multiple phenotypic effects. So phenotypic symptoms of cystic fibrosis are trouble breathing, lung infections, because things are not cleared out of the lungs. With that mucus there, you don't get proper oxygen transport. Sometimes fertility issues, especially in men, trouble growing and gaining weight because you can't absorb nutrients. Often trouble using the bathroom, trouble with bowel movements, greasy stools, things like that. And then another side effect is that the skin tastes salty. So even though we have one specific protein change, you have many phenotypic effects. Another example is sickle cell anemia, or sickle cell disease. This is a single point mutation in a gene that produces hemoglobin in your red blood cells. And so the immediate effect is misshapen hemoglobin. Now this causes reduced oxygen transport because that misshapen hemoglobin produces a misshapen blood cell. It becomes a sickle shape. So normally you have these nice round plump red blood cells. Instead you get this sickle shape. So the immediate effect of the gene is this misshaped or mutated hemoglobin, but you get many different phenotypic effects. So the first is anemia, which is a shortage of red blood cells because those sickle cells die more frequently. And red blood cells normally live much longer, usually about 120 days, but sickle cell blood lives 10 to 20 days. And so the normal turnover, production of new blood, is much slower because the blood cells are dying much faster. And then, of course, without those red blood cells, you don't get the oxygen you need taken to your organs and your tissues. And so this causes fatigue. There's often a lot of pain associated with this disease because your red blood cells block those sickle cell shaped blood cells block blood flow into tiny blood vessels. So there can be pain in the chest, abdomen, and joints, sometimes in the bones. There's often painful swelling of the hands and feet. This is again because the sickle cell shaped blood cells are blocking blood flow. And then you also can have more frequent infections because sickle cell can accumulate in the spleen or damage the spleen, and that leads to reduced ability to fight infection. And then finally, you can have delayed growth because you're not getting the oxygen and nutrients effectively delivered to the different parts of your body. So again, a single specific gene causing a very specific protein change, but having massive effects throughout your entire body. One more that we're going to talk about for this lecture, and this is something called lethal alleles. So basically, in a lethal allele, you end up with a missing phenotype in your offspring. Now, that doesn't mean that individuals don't have that genotype and phenotype. It means they don't survive with it. So let's say we have a wild type allele. Remember, wild type is often referenced by a plus sign. And then our book is labeling the lethal allele as LET for lethal. So if you are homozygous wild type, you're going to be totally normal. If you are heterozygous for the wild type and lethal allele, you'll still survive. If you are homozygous recessive for that lethal allele, you are not going to survive. So in the offspring, if you could have two situations. One, those offspring never develop. So you end up with basically only the homozygous dominant or the heterozygous individual, so only the homozygous phenotype, or they might develop and not survive through to birth. So you could have either thing happening. So if you are homozygous lethal, then 1/4 of the offspring are going to die, which means the remaining 3/4 are normal, but those are the only individuals. So 2/3 will be heterozygous and 1/3 will be homozygous normal. You won't even see that 1/4 homozygous recessive. So if you are expecting normal Mendelian ratios, but you are missing a phenotype in your offspring, you probably have a lethal allele. Generally, lethal alleles are going to be recessive, but they can be dominant. Couple examples of recessive lethal alleles: cystic fibrosis and sickle cell anemia are considered recessive lethals. These are treatable to some extent, but ultimately usually lead in death of the individual when they are homozygous. Tay-Sachs is another one. And then we also have Huntington's disease, which is actually a dominant lethal allele, because individuals that have the dominant allele usually end up dying, and along the way they have a lot of neural degeneration and mental deterioration related to that. Recessive lethal alleles don't cause death in the heterozygote because there's normally some threshold level of protein that needs to be produced. So these individuals are producing enough protein in order to survive like their homozygous normal individuals. So let's look at an example. Curly wings in fruit flies is a dominant lethal allele. So in this one, when the lethal allele is dominant, you still need 2 copies of that lethal in order for it to become that way. So the wild type in this case is actually the recessive. But the homozygous results in the curly phenotype. So we can see the normal and curly, the curly wings here. And then if it's wild type, these are going to be the regular wings that we saw on our fruit fly. It's more like a butterfly. We'll just pretend that wasn't there. All right. The whole point, before I digressed, was that you can also have the dominant lethal. So in this case, you wouldn't see any of those homozygous dominant individuals.